I think we can start. It's a great pleasure to have Alexander Petroff from Clark University. Alex got his bachelor in Carleton College, and then in 2006, and then moved to MIT for his PhD. Since 2011, he moved to Rockefeller University, and after 6 years of postdoctoral, now he's faculty at Clark University. He's working on a variety of topics which could be classified for the lovers of classifications as soft condensed matter physics or biophysics, and today we'll talk about nanoswinging.
Yeah, that's right. Great, it's a pleasure to be here, so nice to meet all y'all. So I'm going to be talking today about some work that I started at Rockefeller in the lab of Albert Liebscher and is now continuing on into my lab at Clark. So basically what I'm interested in, I'm interested in a number of problems about how bacteria and microbial communities organize, with a general thought that if you think about the way that bacteria live, the typical scale of a bacterium and the way that it's interacting with its environment, it's so small and it's manipulating the world on such a small scale that it's often limited by basic physical laws, such as how quickly can diffusion transport nutrients through the water, or how much energy can you get out of burning chemical A with chemical B. And so by understanding what the physical constraints are acting on either on an organism or on an ecosystem, you can put some strong constraints onto how evolution can, force species down specific pathways, or how organisms can interact with one another, and questions like this. So I guess everything that I'm talking about today is about the interaction of a bacterium with a surface. So if you've got a bacterium swimming along, what happens when it slams into a surface? So this problem is generally important for both ecological systems, thinking about how an organism might attach to a surface to avoid being swept away by a flow or position itself in a chemical gradient. It's also important in medical fields. So if you think about plaque forming on your teeth, that's bacteria swimming up and attaching to a surface. And as we'll see, the fluid mechanics associated with a bacterium approaching a surface are quite subtle. And so today what I'm going to be talking about is one particularly fascinating bacterium called Thiovulum majus. I'll introduce you formally in a moment or two. And we'll start by thinking about what happens when a single bacterium swims against a surface, and then we'll think about what happens when groups of bacteria swim against a surface. So what makes my research a little bit unusual is usually when people are studying problems like this, they're studying one of a small number of bacteria, typically Pseudomonas or E. coli, or there are a few domesticated bacterial strains that are sort of the workhorses of biological physics. If you go into nature and if you go into mud in its pure complexity, its pure marvel, its pure messiness, you can find all sorts of magnificent microbes that are absolutely mesmerizing. So the one that we'll be talking about today is called Thiovulum majus, and it's the fastest swimming bacterium in the world. So this is what you see when you look into mud. So this is some mud that we collected from the coast of Massachusetts. And as you look into it, you can see no end of little swirling animalcules and school bus-sized nematodes moving through the environment. And some of the questions we're thinking about are, how are all of these flickering, bizarre things happening? All right. Anyway, so moving on. So the basic system that I'm going to be thinking about today is life in the top few millimeters of sediment. So we live in a world, the atmosphere is about 21% oxygen. And so this oxygen is going to be in the atmosphere. It's going to equilibrate with the water. The oxygen will diffuse from the atmosphere into the water, from the water into the pore spaces within the sediment. And there are organisms all throughout it consuming the oxygen. So given the typical diffusion coefficient of oxygen, sort of 10 to the minus 5 centimeters squared per second for diffusion in free water, and given the typical metabolic rates of microbes, the penetration depth of oxygen into sediment is very small, typically about 1 millimeter, which is shocking, right? We live in 21% oxygen atmosphere, 1 millimeter below the surface, there's no oxygen left. It's all consumed by the microbes on these scales. And so these large scale, by the standards of bacteria, these large-scale chemical gradients make the environment of mud really fascinating. It's, if I'm in this room, the chemical concentration on this side of the room is essentially the same as on that side of the room, and I can walk back and forth without worrying that I'm going to burn to death or lack oxygen or whatever my chemical peculiarities demand of me. So microbes have learned some clever ways of living within these gradients. So if we look up at the surface, so here's the surface of the water, oxygen diffuses down into it, and at about this level here, all of the oxygen has been taken up. I said 1 millimeter, it depends on things like how quickly oxygen is being produced, of order a millimeter. So at this point, all the oxygen has been consumed, and below this, things are still doing metabolism. They're still burning sugars, except instead of using oxygen, they're using a different electron acceptor. They're using sulfur instead of oxygen, and so they produce hydrogen sulfide. So there's a flux of H2S coming from below and a flux of oxygen coming from above. And one group of these bacteria have figured out how to get energy out of burning hydrogen sulfide with oxygen, and that releases some energy, and they live off of this energy. So you've probably seen these sorts of bacteria before. They're most famous for living at deep sea vents. So all that white stuff that's an ecosystem that's completely isolated from the sun. It's their same bacteria, except instead of living several kilometers below the surface of the ocean, they're living a few millimeters below the surface of the sand. And of course, please yell at me and ask me questions and things like that. So if you're one of these bacteria, and you're living at the penetration depth of oxygen, so you're living where the oxygen is just starting to disappear, and you're living where the sulfide is just beginning to meet the oxygen. At this point right here, it's a really nice place for you to live because you've got lots of oxygen to burn with lots of sulfide, and so you've got a continuous flux of oxygen and sulfide coming towards you. And the problem is that the way that it's the mechanism by which these chemicals are being transported to you is by diffusion, which ends up being slow compared to your metabolic rate. So if you were, a normal bacterium like E. coli or something like that, you would have techniques to swim up a chemical gradient. But notice that if you're sitting right here and you swim to a place with more oxygen in it, you lose your sulfide. If you swim to a place with more sulfide, you lose your oxygen. And so you're sort of bound at that interface, but you're limited by how quickly you can get food. So we've got some techniques. So these bacteria that I'm talking about are uncultured. You can't buy them in any store. So if you want to study these bacteria, you need to go get a big bucket full of mud and bring it back to the lab and do some gardening to try to entice these bacteria that I just showed you up out of the water. So we can set up chemical environments that force them up out of the sediment, and you can reach in there with a pipette, grab them out of there, and look at them under a microscope. And you look at these bacterium, this is one of them. So this is the T. majus that I was mentioning earlier. It's a very unusual bacteria in a few different ways. So this particular sample, we dried out a whole bunch to look at under the electron microscope. Typically, the radius of these is about 4.5 micron. For reference, most bacteria are about 1 micron. So it's very big. What's really unusual, though, is if you look at all these little white filaments around it, so each one of these is what's called a flagellum. So this is a corkscrew-shaped protein, and if the cell rotates this protein through the water, it exerts a point force on the fluid, and it can push the fluid away, and so force balance on the cell pushes the cell through the water. So these flagella are how these bacteria swim through the water. So E. coli, most bacteria that you would ever expect to meet have 1 or 2 or 5 or at most 20 or so flagella. This fella has 200 flagella, typically. So it has an obscene number of flagella. So why does it have so many? So here's what the cell looks like in real life. So this is under the microscope. And it's also got another, evolutionary superpower. So it can exude a little piece of mucus. You can't see it at this magnification, so I drew it in with a line. So it's tied in place to a point. And so once it's tied off, it can rotate its flagella, and you can see that it creates a fluid flow. So this is a cell that, rather than swimming through the water to get more nutrients, it's creating a fluid flow that's pulling water towards it. So If you do particle tracking, you can figure out what the velocity field looks like. It looks an awful lot like a force dipole. Yes?
Is that in real time?
Yes, it is. Yeah. It's almost real time. I think I may have slowed it down by like 30% or something. So it's very close to real time. Sorry? Okay. Cool. So you can look at the flow fields that it's creating. So it's actively creating a flow. So if you measure the force, so for example, you can track the speed of these particles. You can say, I've got a point force. I solve the Stokes equations around that to find the fluid flow. So given the velocity at a point, I know the speed at which, I know the force that the cell is exerting on the fluid. So you can infer the force that the cell is exerting, and it's about 40 piconewtons, which is an order of magnitude greater than what bacteria typically exert on the fluid. That's maybe not remarkable since it's got an order of magnitude more flagella than you would expect, but it's still curious about why is this cell exerting such a large force? So to understand this, we can think about this from the cell's perspective of, I'm a cell sitting here, and I'm trying to pull oxygenated water from the far wall. So I create a fluid flow that transports that oxygen towards me. So there's a lot of oxygen at that far wall, very little oxygen close to me. So as I pull that water towards me, the oxygen, the parcel of water that I'm pulling towards me, is diffusing out into the surrounding anoxic water. So if I take a long time pulling that fluid towards me, by the time the water gets to me, it's equilibrated with the anoxic fluid around it, which means that I get no oxygen out of it. So there has to be some typical speed that you need to pull towards it. And you can figure that out quite easily by defining the Peclet number as the ratio of the advective flux. So this is the flux of oxygen coming towards me. That's a result of the fluid flow that I'm creating. And you can compare that to the diffusive flux. And so blah, 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 you solve the Stokes equations, and you can find that the Peclet number is the force the cell is exerting, divided by a typical number, which is determined by the diffusion coefficient of oxygen in water and the viscosity of water. So if you think about what the cell can manipulate, so basically, if this strategy is to make any sense, this number has to be at least of order 1. Otherwise, all the oxygen is gone. So the cell needs to make this number of order 1. It can't change the diffusion coefficient of oxygen in water, and it can't change the viscosity of water. So the only thing that evolution can allow it to do is to adjust the force that it exerts. So the diffusion coefficient and the viscosity of water define a typical force the cell needs to exert to benefit it all from this strategy. It ends up being about 25 piconewton, which is similar to the force that the cell is actually exerting. So a little bit of fluid mechanics, a little bit of Fick's law for diffusion, and you can understand something quantitative about the anatomy of these bacteria. The focus of today's talk is I'm sorry. When the cell is, so I like showing that video, the cell stuck into place. The tether that's holding it into place breaks from time to time, once every couple of minutes, and then the cell goes shooting off. As soon as that tether breaks, the cell goes swimming off at about 600 micron per second, which makes it tied for the fastest bacteria in the world. And so it's interesting to note, though, that the reason that it's such a fast swimmer isn't to try to get from point A to point B fastest. It's so that when it's stuck in place, it can create a large enough flow to actually, physically modify its chemical environment. Yes?
What's the range of motion this flagellum has? It's corkscrew motion?
Yes, so each flagellum is an individual corkscrew, and it can rotate that. That exerts a point force on the fluid. So it's a little bit like a mini propeller. Propellers require, so a propeller on a speedboat requires a high Reynolds number flow. So these are specifically low Reynolds number. So the way that it exerts a force is the fact that the drag coefficient of a slender object is different for flow parallel to it versus perpendicular. And so you can work out that as you rotate a corkscrew-shaped object through it, the net drag on it has a net direction that's along the axis of the helix.
Does it coordinate like circular motion? Does it turn around by kind of coordinating the different?
So nobody knows. So this is one of the exciting things about working with these uncultured bacteria. The typical answer to most fundamental questions that you're going to ask me are nobody knows. So the nice thing with E. coli is that you can use fluorescent tags in the flagella to stain them and to be able to actually look at these flagella. So I can see the 200 flagella under the electron microscope, but figuring out how these flagella bind up to exert such a large force, it's totally mysterious. There are a bunch of peculiarities with the flagella of these cells. So the question that I need to be focused on today is, I've got a fast-swimming bacterium. What happens when it hits a wall? So let's watch it. So we've got a cell. This is, of course, a cartoon. We can imagine it swims down, it hits the wall, and it might bounce off of it. And so we can build a microfluidic chamber, load it up with bacteria, and watch the bacteria hit the wall and see what happens. So here's an example of that. The cell comes flying down, it hits the wall. So this blue is the wall. And you can see that it came down along this track. It hit the wall at this point, and then bounced off of it. So something that you'll notice right away is that this isn't like throwing a ball against the ground. So if you throw a ball against the ground, you've got conservation of momentum, which means that the angle coming in is equal to the angle coming out. So this is a cell that's submerged in a viscous fluid. Moment by moment, the force that it's exerting is exactly balanced by drag. The momentum of the cell at each instant is diffusing instantaneously into the surrounding fluid. So momentum is not conserved here. And so you can see that these angles are not necessarily the same. Yes?
Another way of saying this, like, inertia is not important.
Yeah, the cell, that's right, yes, there's no inertia. Low Reynolds number, the entire system is overdamped. And so just like, the mass doesn't matter, yeah. Yes, no inertia. Aristotle would be pleased. So when these cells swim in and hit the wall, they don't necessarily bounce off as I just showed you. So here's another video. The cell comes swimming down, it hits the wall, and it's stuck in place. And so you can see it wiggling back and forth. It moves minutely over the surface. And it's just going to be stuck here for a moment or two. This is real time. And then it goes shooting off again. So this is a case of a cell swimming down. Somehow there's a stable or there's a stable fixed point of a cell swimming towards a wall. You can imagine this a little bit like a fly trying to escape through a closed window. It's pressed up against the wall trying to move forward, but the wall is resisting it. So you can see that there are these two types of motion. So let's try to pick them apart a little bit. I should also keep an eye on the time. All right, so let's begin with what happens when the cell is swimming towards the surface. So here's the cell. It's swimming in this direction. So the cell is pushing water with its flagella backwards. That exerts a net force on the cell body. So the cell is moving forward. As the cell pushes through the fluid, it pushes water ahead of it. And so conservation of mass requires that there be a flux of water in from the sides that goes out in front of it to represent the water the cell is pushing out ahead of it, and an equal flow coming backwards. So the cell is moving at a constant velocity. There's no net force. The force exerted by the flagellum is balanced by drag on the cell. And so it's creating a flow that pushes water ahead of it. So you can imagine that as it's moving towards the wall, it's pushing water towards the wall. It's pulling water in from the sides. So as the cell approaches the wall, the flow that it's creating tends to turn the cell to swim parallel to the wall. If you want to analyze this motion very nicely, so what we're solving is grad squared u or if you like, the Laplacian of pressure is 0. So what we're solving here is not so dissimilar from thinking about the electric field around a point charge. So just like you can set up an image charge to figure out the motion of an electron near a conductor, You can do the same thing with a cell and have an image bacterium on the other side to satisfy the no-slip boundary condition on the wall. And then you can think about how this cell moves towards the wall as it's being advected by its image, by the image cell. In any case, the predictions of the Stokes equations are that as this approaches the wall, it turns to swim parallel to it. And we can measure this experimentally. So this is the distance the cell is from the wall, distance divided by the body size of the cell. And this is how quickly it's turning. So when it's more than about 2 body lengths away from the wall, it's swimming in a straight line. Once it gets within about 2 body lengths, it begins to rotate to turn parallel to the wall. So that's fair enough, but it makes us even more curious about how it is that the cells could ever stick to the surface. So right now we have an image of a cell coming in, it turns, and we'd expect it to swim parallel to the wall. This is what E. coli do.
This may be the same as Marcelo's question. When you started by saying he's stuck to a rope, he's pulling the rope towards him and it breaks, right?
Uh-huh.
This picture in my head that he's moving forward by pulling the water towards him. But in this picture,
it's pushing water behind it, yeah. So the trajectory of the cells and the flow fields, as best we can work it out, make it so there are both types of bacteria in the world, some that push water from behind it and some that pull water towards it. And both of them exist, both of them are good at swimming. The flow fields that these create seem to be a little bit more similar to the pusher types. But that being said, you know, the difference between pushing and pulling is coming down to you know, thinking about a single flagella or a small number of flagella. This thing is covered with them. So it's probably fair to just say it's both pushing and pulling on the fluid. And so perhaps the mystery of how it gets stuck to the surface is maybe not so mysterious. Yes?
Question. Does the time the bacteria gets stuck on the wall depend on the angle of incidence?
Yes. Yeah. Uh-huh. So I'll show some data in a moment. So this is justifying the explanation for why a cell, as it swims towards the wall, would turn parallel to it, as we see. We can also imagine that something else takes over. Ooh. No, that's reckless. All right. So we can also imagine, let's also try to think about what the fluid forces are acting on the cell when it's close to a wall. So here we have a cell. It's very close to the wall now. It's exerting a force in this direction. So the flagella is pushing the cell. So there's a component to the force that pushes the cell tangent to the surface. So this cell is going to try to push itself laterally over the surface. So that means there has to be a fluid flow above the cell as it's pulling the water along with it, and a fluid flow below the cell. So within this tiny gap between the cell body and the wall, we need to have the velocity of the water going from 0 at the fixed boundary to whatever the motion of the cell is. So the small gap between the cell and the wall compared to the huge gap between the cell and, you know, infinity means that as you push the cell over this way, the drag at the base of it is much larger than the drag at the top of it. And so as the cell gets pushed in this direction, there's a net drag force in this direction over here. And so there's a net torque that causes the cell to be, as it gets pushed over, the torque on the bottom moves more slowly than the top, and so the cell tends to right itself. So sort of sketching it out in our mind, we can kind of imagine that there's a stable fixed point for the cell oriented normal to the surface. And if you work out the Stokes equations, there indeed is. So the way so putting these two together, we know that when you're far away from a surface, the fixed point at theta is equal to 0 is unstable. So as you move towards it, any deviation, you tend to move parallel to it. If you're close to the wall, then theta equals 0 is a stable fixed point. So we need to have so we've got a fixed point, and we know that it should turn from an unstable fixed point into a stable fixed point at some critical distance from the wall. And so the simplest way that you can work this out is to think about a subcritical pitchfork bifurcation in the distance of the cell from the wall. So as you approach the wall, the fixed point at theta is equal to 0 goes from unstable to stable below some critical length. I'm going to rush a little bit because there's a bunch of other stuff I want to talk about. So you were asking about how does the incoming angle influence the rate at which it gets caught. So it's experimentally easier to measure the trapping probability rather than the time that it's stuck, for the simple reason that we're filming a bacterium next to a wall and they're static, and our image processing software tends to lose the bacteria after some tens of seconds. You know, it's just hard to track a tiny dot under a microscope for a very long time. So we have trouble imaging the bacteria, measuring the. So our estimates of the time that they're stuck is biased towards the ones that are stuck for the smallest amount of time, simply because it's easier to keep track of them. So it's much easier to measure the trapping probability. And you see quite nicely that the probability that a cell gets trapped decreases exponentially with the angle at which it approaches the wall. So the idea here is, as the cell is coming towards the wall, it turns towards to swim parallel to the surface. If it's coming in at a very narrow angle, it turns, and as it hits the surface, it's still within the basin of attraction of the stable fixed point that we believe is there. If it's coming in at a steep angle, it turns and remains outside of the basin of attraction, and so can escape. Just to quickly justify the idea that there is a stable fixed point, we want to be able to measure the rate at which the bacteria stabilizes to that stable fixed point. So we can do this indirectly. So in the videos that we can see two different types of motion. We can see the cells come in, hit the wall, and bounce out at the same angle. And we can see cells do the, absolutely, if momentum were conserved, it would absolutely be unbelievable. The cell comes in and it bounces out at an angle greater at a negative reflection angle. So what we're going to do is we can measure the probability that the cell continues in its same direction. So what is the dynamical memory of the system as it moves close to this fixed point? So we can measure the probability that it continues in the same direction and the probability that it gets reflected back, and we can measure the entropy of that. So basically what we're saying is, given that you came in from let's say the right, how much do I know about the direction that you'll be leaving the wall from? And so if you measure the entropy as a function of the trapping time, you can see that it increases up to about 0.2 seconds. So basically what's happening is, after about 200 milliseconds, the fact that the bacterium came in from the right tells you nothing about the direction that it will be moving away from it. So it seems that as a cell gets close to the surface, it takes about 200 milliseconds for it to move to that stable fixed point. And once it's at that stable fixed point, the dynamics by which it leaves are symmetric. And just looking at the ways that the all right, cool. Just looking at the distribution of angles at which the bacteria leave the surface. In red, we have the free swimming cells, so the ones that did not get trapped. Remarkably, regardless of the angle that they came in from, they all leave at the same angle. If they're trapped, they still leave at that same angle, but they're equally likely to leave in the positive direction or in the negative direction. I may have done that too quickly. Folks good? All right, cool. I'll keep going. So all of that was just about a single bacterium colliding with a wall, and that's the sort of active research. I'm not convinced about that subcritical pitchfork bifurcation. That's the sort of active problem that we're working on, about trying to understand their fluid dynamics. If you do any of the simplest stuff for a single bacterium with a single flagellum, nothing I said makes sense, and so we're missing something interesting, and I think that it's related to the fact that we have many, many flagella. But that will be for later. In any case, so moving on to the next part, now we're going to be thinking more about their trapped cells. So I was looking at that from the side. So that was a large container that's thin to make it very easy to watch the bacteria as they swim up towards the wall and bounce off of it and measure the angles coming in and out of it. So now what I want to focus on are the cells that are stuck, that are dynamically trapped to the boundary. So I changed geometries, and now I have a microscope slide and a 300 micron spacer, and then a cover slip, and I'm just going to fill up the fluid in between with as many of these free-swimming bacteria as I can get. So let's watch them get trapped. So this is now focused on the top piece of glass. So this, and this are all bacteria that are trying to swim towards you. I drew a red dot here where a new bacterium is about to collide with the surface. So a cell swims up, it bangs into it, and you can see that it can move laterally over the surface. And if it gets close to one of the other bound cells, they trap one another and they rotate about each other. Not obvious why. Maybe it is obvious. Yes?
Spinning around their axis.
Yes, they are. So going back to, I want white chalk. Yes, that should be visible. So we've got a cell, we've got the flagella. The flagella turn in a given direction. So the flagella rotates in one direction. So the cell is exerting a net torque on the surrounding fluid, which means that, so torque balance on the cell requires that the cell rotate in the opposite direction. So the flagella rotate in one direction, the cell body rotates in the opposite direction, just like a helicopter would if you chopped off the back of it. Cool. All right, so we've got a couple of different components to this motion. We've got the cell, it collides with a wall. So I hope I've convinced you that we at least roughly understand how the cell gets stuck. Then it can move laterally over the surface, and then it can get bound to a neighbor. So let's break apart these different pieces. So let's start with how the cell moves laterally over the surface. So continuing our same argument about a stable fixed point as a cell approaches the surface. So it moves laterally. So if the cell isn't oriented normal to the surface, there's a lateral force on the cell which pushes it over the surface, and it moves upwards. So the other important thing to recognize about these flagella is that they're good, but they're not great, and they're hard to coordinate. So as these cells are rotating its flagella, it's not guaranteed that every single flagellum is actually going to rotate. You know, you have to be moving protons through the cell wall in order to make these rotate. Sometimes they get stuck, and so the force that the cell is trying to exert in a given direction is not what it ends up exerting. Flagella can bang into stuff, and so as these cells are exerting a force in this direction, there's also a rotational diffusion. So basically what's happening is the cell is misaligned from the surface, it gets pushed over, and then it gets effectively thermal kicks from all the misfiring from all the misfiring flagella. So there's the dynamics of the cell orienting normal to the surface, plus some stochastic dynamics associated with flagella misfiring. So if we decompose the motion of the cell, so the cell is rotating like a top, as we have here. It's pushing towards the surface. It's precessing about its axis as a result of the rotation of the flagella. And so I don't know how well you can see blue on black, but the cell is making these tiny little circles, sort of drifting in some direction. If you measure the direction the cell is going as a function of time, you can see that theta is constantly increasing. So it's tracing out these little circles. So to decompose the precession of the cell from the general drift, you can do a Poincaré section and measure the position as it's moving in the, let's say, x direction. So if you look at all of this, you can measure the distribution of velocities. This is the logarithm of the probability. As long as the velocity is relatively small, you're not far from Gaussian. So it's getting little Gaussian kicks from its flagella. You measure the mean squared displacement of the cell as a function of time. It's moving very nicely like a Brownian particle. So the typical diffusion coefficient you can measure. So basically what we have now are a bunch of individual cells that are diffusing in 2 dimensions. So you can sort of imagine them like a 2D gas. This typical diffusion coefficient, well, let's keep in mind that this entire system is far from equilibrium. These cells are moving about as a result of actively burning sulfide with oxygen, using that energy to rotate their flagella, and so there's a flux of energy into the cell that's getting dissipated by the motion. So this system is far from equilibrium. Given the size of the cell and given this diffusion coefficient, you can figure out what the effective temperature is. How high would the temperature need to be for a particle of this size to diffuse with this diffusion coefficient? And it's like 40,000 degrees Kelvin. So these cells are diffusing like particles, but far from equilibrium. Next, let's think about how it is that the cells attract one another. We saw there's 2 cells get stuck together. Yes?
Just quickly, so this rotational diffusion coefficient, even if it's being bombarded by water molecules, that will give a rotation.
Yeah, so the
Smaller diffusion coefficient.
Yeah, so that would be the rotational diffusion coefficient that you would get from the from KBT fluctuations in the fluid.
So the temperature is then room temperature in that case.
It is, sorry?
So effectively the temperature would be room temperature.
Yeah, exactly, yes.
Much less.
Yeah. Yeah, the lab got hot, but never that hot. So let's think about the flow of water around one of these bound bacteria. So here's the cell. It's pushing fluid up away from the surface. So it's pushing water up away from the surface. Here's the cell. It's pushing water in this direction. Conservation of mass requires that there be a flow over the surface towards the cell and then up and away. Additionally, as we've discussed, the rotation of the flagella in one direction causes the cell body to rotate in the opposite direction. So the water gets pulled towards the cell and then around it and away. Sorry, posterity. Okay, that still works? Good. All right, so if you look at, so you can solve for the flow field around such a bound bacterium. And so these black lines show the velocity field cut through the equator of the cell. So the cell is creating a flow that in 2 dimensions makes it look like a point sink. So every cell looks like a point sink. So it's perhaps natural that these cells would attract one another. So here's one cell, it's creating a flow of water around it. Here's another cell, it's diffusing over the surface, and it's being advected by the flow that this cell is creating. So we could figure out, given the flow that this is creating, so you can set up the problem with there's image bacteria, as I discussed, to figure out what the velocity field around a bound cell is. It decreases like 1/distance to the 4th. The prefactor depends on the size of the cell, the viscosity of water, and the force the cell is exerting. So we know all of those. And so we can figure out what the velocity is at a given distance from the cell. So presumably these cells should get bound when the velocity of the fluid that a cell is inducing is comparable to the velocity fluctuations that we measure as a result of Brownian motion. So if you balance the flow that the cell is exerting compared to the typical velocity fluctuations of a bound cell, you can find that these two become comparable when they're separated by about 9 microns. 1/distance to the 4th decreases really quickly, right? So if this cell is more than a few body lengths away, the velocity field is is negligible. And so we can think about these bacteria freely diffusing in 2 dimensions. And if they diffuse within a given distance of about 9 micron, we expect them to get stuck together. So we can measure that. So if we think about 2 cells that are randomly diffusing, we can measure how far apart they need to be before they get stuck by looking in the data and seeing, all right, I saw 2 cells get stuck at a time tc. How far apart were they 1 second before that? So no interaction between the cells, just saying they're randomly moving. If 2 cells are randomly moving and they hit each other at time is equal to 0, 1 second before that, they probably weren't a meter apart, because that would take a lot of unlikely events to push them together 1 second later. And they probably weren't separated by 100 nanometers, because then they would have hit one another long before a second. So given that they hit each other at a second, how far apart were they before that? So this is two random. This is the how long does it take two Brownian particles to collide? So you can work it out. The expected distance between them should increase like the square root of time, the effective diffusion coefficient of the two, and this 16 is related to, you've got two cells diffusing in 2 dimensions. And so what we see are the cells, so the blue line here is data, the red line is exactly this function, so the distance between them decreases like the square root of time until they get within 13 micron of one another, and then they go rapidly to 0. So the cells are, so as soon as the cells get within about 13 micron of one another, they get pulled together into these dimers. So that's 2 cells. What about more cells? So there's nothing to stop that from having cells pile up and more cells get stuck together. And so this is 1, 2, 3, 4, 5, 6, 7, 8 cells that are all stuck together. And if we watch them, they all rotate like a little crystal. So first of all, we can understand the packing. We've got mutual attraction between cells. So the stable organizations are going to be the ones that get them as close together as possible. The maximum packing for 2D spheres is in a hexagonal lattice. So we get a hexagonal lattice of cells. The chirality of the flagella is determined genetically. So all of these cells rotate in the same direction. And so we have a bunch of cells all rotating in the same direction and all rotating about one another. So there's 8 cells. Here's about 127. And you can see a lot going on here. You can see portions of the group sort of rotating as fixed bodies, solid crystalline grains, falling over the surface of a larger crystal. You can see this little group get pulled from one crystal into the next. Lots going on. And here are some gigantic ones. So here are hundreds of cells with lots of tiny crystals actively dancing within them. Yes?
What's underneath it?
A lot of bacteria. So what happens is these cells tend to hang out at about 4% atmospheric oxygen. So remember that these are the bacteria that live right at the penetration depth of oxygen. If you expose them to 100% oxygen, the free oxygen just burns them to death and they die. If you expose them to 0% oxygen, they suffocate and die. So these are delicate little flowers, and they're very good at organizing themselves. So if you take a bunch of free fluid and mix it up and put it into a container, what happens is you have cells burning oxygen at different rates, creating where cells happen to be a little bit more dense. They consume oxygen more quickly, so the oxygen level there falls. And so cells tend to pile up within these little anoxic regions near the borders of it. And so this is a high density of cells. We're only focused on the top plate, but there are 1,000 times more free swimming cells out of the focal plane, and that's who you see swimming all around here. And they're swimming here as opposed to here, because this has less oxygen in it than this, because a careless postdoc was too slow filling the chamber. That was me.
Okay. And
Yes.
nice question. How do they know where to go for the right amount of oxygen?
So again, I'll answer most honestly with we don't really know. The sort of next point there is bacteria have lots of different ways of sensing their local chemical environment. You can build oxygen sensors that tie into signaling networks within the cell. And these cells have a bunch of the normal chemotactic pathways. So the way that E. coli knows which way to go is E. coli is very small, and it's much smaller than the typical gradients, and in fact too small to measure the difference in concentration between one side and the other. So E. coli sits in a place, measures the oxygen concentration, takes a random step in some direction and compares the two, and then it adjusts the distance that it swims between one measurement and the next, based off of if the concentration here is greater than or less than the concentration here. So if you look at the cell, it does a biased random walk up a chemical gradient. These cells have the same biochemical signaling pathways. They move more smoothly, and it's not clear why. I can speculate, but it's not substantiated, and it would take a while. So we can talk more about that later afterwards. So one of the things that you'll notice with all these crystals that I just showed you are that first one. So all these videos were slowed down by, all these were at 2/3 speed. So that first crystal I showed you made 3 full rotations in the video length. The final huge crystal is hardly rotated at all. So if you measure the rotation period, it increases proportional to the number of bacteria within the crystal. Why might that be? So you can write down force balance on each cell. So this is balancing viscous force with the force of each cell. So each cell rotates with angular velocity omega ij. The rotational component of the flow field decays more quickly than the normal component. So so you can approximate it as every cell rotates about its neighbors. So we've got rotation about your neighbors. Cij here is the adjacency matrix for these networks. We've got mutual attraction between all of the cells that decreases like 1/4 distance to the 4th, and then they can't overlap one another. And if you non-dimensionalize, you get a single dimensionless parameter, which is the dimensionless rotation rate. So how quickly you're rotating compared to how quickly you're pulling fluid towards you. Another good way of seeing this is gamma is the ratio of the typical typical shear forces exerted by one cell on its neighbors compared to the typical compressive forces. So you can write down force balance. And so if we think about one of these little crystals, we have all the cells rotating in the same direction. So if we think about the net force acting on each cell, as this cell rotates, it pushes this cell downwards. As this cell rotates, it pushes this cell upwards. And so their effects cancel out. So if we figure out which cells experience a net force, it's exactly Stokes' theorem, right? This is the integral of the curl, is the line integral around the boundary. So the net force acting on this is only due to cells on the boundary or cells along topological defects within the crystal. So if you do the force balance, so each cell, so the force acting on the cell is proportional to the perimeter. So square root of the number times the radius of gyration gives you another square root n. So the typical torque is proportional to the number of cells within it. And then you can work out what the viscous torque is associated with taking a disk and rotating it through the fluid. So the viscous torque is proportional to the angular velocity times the number of cells squared. So balancing the torque generated by the cells to the viscous torque, you can solve for omega, and you get how the period depends on the number of cells, and it works reasonably well. So yeah, this will work. Good. It's fun to watch more of these cells. You can see, so all of these cells are rotating in the clockwise direction. The frame rate is a little bit low. So if you see a cell rotating in the wrong direction, the video has been, there's aliasing problems. So you can see If you look within this, cells within the center of it stay more or less fixed relative to their neighbors. They're simply slowly pulled through the fluid as the crystal rotates. When we look at the surface, cells can evaporate off of the surface, and you can see that they trigger little avalanches behind them. So you can work out the stability criteria. If you read our paper, we work out the stability in that. Essentially, the stability criterion is exactly the same as what you get out of Coulomb failure, where if I have a dense packing of spheres, if I want to move one line parallel to another, so I'm pushing them laterally, I need to pull them out. I need to make the crystal a little bit less dense at a point. And so basically what you end up with is shear forces need to be greater than normal forces by a factor of something not unlike cosine 60 degrees from the dense packing. And so you can work out exactly what the stability criterion is. And yeah. So if you look at small crystals, so these crystals are taking their shape as a result of bacteria evaporating from the surface, being pulled by one another, and moving through the fluid. So if we watch these, you can ask, within the set of these dynamics, what sort of fixed points can we expect? It's remarkable to watch them go as cells evaporate from the surface as they move past one another. You can see that initial unusual shape slowly converged into this now stable, faceted structure, which deserves some thought. And it's stable for a good long while. So let's think about the sorts of shapes that we can expect. So now, okay, so biology is done. Everything after this is the ravings of my own mind. So we're just going to solve this numerically and figure out what sorts of behaviors we should expect. So let's think about how the geometry of a crystal is dependent on the shear parameter, so the ratio of shear to compressive forces, and what happens if that varies a little bit. So let's play. So let's start off with 8 cells, and according to this game, what we're going to do is start off at a reasonably low value of gamma, and I mean to increase it temporarily, and then I mean to keep it at a high level, and I mean to drop gamma back down low again. And let's watch to see what happens. So at low gamma, everybody gets pulled together. As I increase gamma, I'm increasing shear forces, so the whole thing rotates faster and faster. At some point, it becomes unstable. And maybe you might say it melted. As it rearranges itself, you can see that it finds another stable organization. As I decrease gamma again, it remains stable. So there's something non-reversible about this. So, trained in physics, we see a bunch of crazy things happening. I associated these earlier with a 2D gas, and so it's natural to think about one of these things falling apart as it melting. And that's wrong. There's no noise anywhere in this. So what we're seeing here is at a low value of gamma, the system is brought towards one stable fixed point. As I increase gamma, that fixed point becomes unstable, and now it's moving through the space. So this is 8 cells moving in 2 dimensions. So that's 16 dimensions. We've got 2 translational symmetries and a rotational symmetry. So it's moving through a 13-dimensional space. So within that 13-dimensional space, at low gamma, we started off in the basin of attraction of this organization. As I increase gamma, this becomes unstable, and it wanders through this 13-dimensional space before it hits a new stable fixed point. And then as I decrease gamma again, that remains stable. The facets are important for these. So if I start off at a very high value of gamma, if I have a special number of cells, 1, 7, 19, 37, a hex number, I can form a perfectly faceted structure. And if I start off at a high value of gamma, if I add a cell, it instantaneously destabilizes it. And so within this parameter space, I've got a stable core, and then a periodic solution. I've got a periodic orbit. So presumably these dynamics, in addition to allowing stable and unstable fixed points, also allow limit cycles. And we were happy to see at least one limit cycle under the microscope as it wanders about. So, very last slide. So everything I did here was based off of cells rotating in the same direction. A natural question is what would happen if you could engineer these cells to rotate in opposite directions? So red rotates in the clockwise direction, blue in the counterclockwise. If you integrate this for a long time, you can see that the two faces separate. And eventually, so you get pretty good phase separation. And then you integrate for a while longer. And so the entire organization finds a new stable organization, which is two co-rotating gears with a little ratchet. So I've gone over, so I'll ignore all of that. This is my group. So it's fun to be in a physics department now and have people with buckets full of mud. So it's bizarre and amusing. At least I think it is. Anyway, thank you very much.
Question?
So you talked about how these have hundreds of flagella on them. I guess I'm a little confused about the orientation of those. Do they all coordinate to force in the same direction, or do only some of them activate at the same time? It seems like if they're randomly distributed, you wouldn't get any.
Yeah, I'm also confused by it.
Okay. Cool.
Yeah, they do a very good job of exerting a very large force in a specific direction. If you, the flagella are more or less distributed uniformly. There's not any clear preference for one pole or the other. If you look at the other bacteria, Ovobacter, it's funny, both of the fastest bacteria are both named after eggs. Thiovulum is the little sulfur egg, Ovobacter is the egg bacterium. So I don't know who was up to that, but if you look at the other bacteria, if you look at the distribution of its flagellas, they're very nicely ordered. And it's sort of, and you stare at it, and there's some sort of intuition that this cell should swim quickly. If you look at T. majus, it's a mess, and yet it does the same. So there's something funny going on about how cells form bundles, or something peculiar is going on, and nobody knows what it is.
So you said that which way the flagella goes is genetically determined.
Mm-hmm.
So for a single guy,
Yes.
he's got 200 of them.
Yeah.
Are all his 200 the same sense, or does he have a combination of some left ones and some right ones?
A good question.
You can't resolve that in your imaging?
No, I can't. No. I've got a friend who is good at flagellar staining. So a problem with the flagellar stains that at least I know about are they're typically pretty toxic. So if you stain the cells to look at the flagella, you end up looking at a dead bacterium. So my friend has a very clever way of holding a bacterium in place and giving a little puff of the dye so you can get an instantaneous view of it. And I think that would be a lovely prospect to study these bacteria, but we haven't done it.
In your model, you said that there was only one dimensional parameter, gamma.
Yeah.
And was that determined by F naught?
It's determined by the angular velocity, the force that they're exerting, and the drag coefficient.
Gotcha. How reasonable is it to assume that F naught is a constant and doesn't vary from cell to cell?
So They generally swim at about the same speed, which suggests that F naught is similar. I think it might be unrealistic to say that a cell that's surrounded on all sides by 6 others, presumably a bunch of its flagella are getting blocked, so maybe its effective velocity, its effective force is smaller. But we can't measure it with that resolution, so we say it's a constant and press enter.
I have one. I was expecting more interplay between Markovianity and non-Markovianity in this system because at the beginning you were showing this lack of memory effect in the system, but then you have described everything in terms of Markovian Brownian motion, basically.
Yeah.
At which point you can try to see the difference between the two regimes in these systems?
Oh, I think it comes down to there's memory in the system before the cell has enough time to organize to its stable fixed point. And once you're at that stable fixed point, all you're seeing are the Brownian kicks that push you about that, and then you return back towards it. If you come in at a steep enough angle, you don't have enough time to move to that fixed point, and so the dynamics have memory in how long it takes you to turn, and whether or not you fall within that basin of attraction or not. So I think the stability of that fixed point kills the memory of the system.
We can continue the discussion during the coffee and tea break together. Thank you.
Thank you very much.